Hello everyone, I hope you are doing well. Today I'm going to discuss very important topic which is drug receptor interaction and pharmacodynamics. As we know, pharmacodynamic is about the action of a drug on the body and the influence of the drug concentration on the magnitude of the response. Most drugs exert their effect both beneficial and harmful by interacting with the receptors present on the cell surface within the cell. The drug receptor complex initiates alteration in biochemical or a molecular activity of the cell by a process called signal transduction. Now we see what is signal transduction. Drug act as signal and their receptor act as signal detectors. Many receptors signal their recognition of a bound ligand by initiating a series of reactions that ultimately result in a specific intracellular response. Now I move toward the drug receptor complex. This concept is closely related to the formation of complex between enzyme and substrate and antigen antibody. In this cell have different types of receptors, each of which is specific for a particular ligand and produces a unique response. For example, heart contain a membrane receptor that bind and respond to epinephrine or norepinephrine as well as muscarinic receptors specific for acetylcholine. These receptors dynamically interact to control the heart's vital function. The magnitude of the response is proportional to the number of the drug receptor complexes. Here we have an equation where we can say drug plus receptor show drug receptor complex and ultimate biological effect. These receptors not only has the ability to recognize a ligand but can also couple or transduce this binding into a response by causing a conformational change or a biochemical effect. Most receptors are named to indicate the type of a drug, a chemical that interacts with it. For example, the receptor for histamine called histamine receptors. It is important to be aware of that not all the drugs exerts their effect by reacting with the receptors. Now I move toward the receptor states. We have two types of state, that is active state or inactive states. These two states are reversible equilibrium with one another. For example, drugs acting as agonists bind to active state of the receptor and thus rapidly shift the equilibrium from inactive to active. Other drugs may induce a change that may be different from the endogenous ligand. These changes render the receptors less functional or non-functional. Now I have major receptor families. These includes enzymes, nucleic acids, and structural proteins can be considered to be a pharmacologic receptors. However, the richest source of the therapeutically exploitable pharmacologic receptors are protein that are responsible for transducing extracellular signal into intracellular responses. These are four types of receptors. These are ligand gated G protein coupled receptors, enzyme linked receptors, and intracellular receptors. One by one I will go in detail. Now I start with transmembrane ligand gated ion channel, which are responsible for the flow of ions across the cell membrane. The activity of these channel is regulated by the binding of a ligand to the channel. Response to these receptor is very rapid, enduring for only a few milliseconds. These receptors mediate diverse functions including neurotransmission, cardiac conduction, and muscle contraction. For example, stimulation of the nicotinic receptor by acetylcholine results in sodium influx, generation of an action potential, and activation of concentration in skeletal muscle. Benzodiazepine, on the other hand, enhance the stimulation of the gamma aminobutyric acid GABA receptor, resulting in increased chloride influx and hyperpolarization of respective cell. Although not ligand-gated ion channels such as the voltage-gated sodium channel are important drug receptors for several drug classes including local anesthetics. Now I move towards the second receptor type, that is transmembrane G-coupled receptors. These receptors comprise a single alpha helical peptide that has 7 membrane spanning region. The extracellular domain of this receptor usually contains the ligand binding area. A few ligands interact within the receptor transmembrane domain. Intracellularly, these receptors are linked into a G protein, that is GS, GI, and others, having three subunits: an alpha subunit that binds guanosine triphosphate (GTP) and beta gamma subunit. Binding of the appropriate ligand to the extracellular region of the receptor activates the G protein so that guanosine triphosphate replace guanosine diphosphate (GDP) on the alpha subunit. Dissociation of the G protein occurs and both the alpha GTP subunit and the beta gamma subunit subsequently interact with each other cellular effectors, usually an enzyme, a protein, or an ion channel. These effectors then activate second messenger that are responsible for further actions within the cell. Stimulation of these receptors result in responses that last several seconds to minutes. G protein coupled receptors are the most abundant type of the receptors, and their activation accounts for the action of the most therapeutic agent. Important process mediated by G protein coupled receptors including neurotransmission, olfaction, and VN. Now we move towards the second messenger. These are the essential in conducting and amplifying signals coming from the G protein coupled receptors. A common pathway turned on by GS and the other types of the G protein is the activation of adenyl cyclase by alpha GTP subunit, which results in the production of cyclic adenosine monophosphate, a second messenger that regulates protein phosphorylation. G proteins also activate phospholipase C, which is responsible for the generation of two other second messengers, namely inositosol 1,4,5 triphosphate and diacylglycerol. Inositosol 1,4,5 triphosphate is responsible for the regulation of intracellular free calcium concentration and of other protein, as well as diacylglycerol activates several enzymes such as protein kinase within the cell, leading to myriad of physiological effect. Now I have third type of receptor, that is enzymatic receptors. In this type consists of a protein that spans the membrane once and may form dimers or a multi-subunit complexes. These receptors also have cytosolic enzyme activity as an integral component of their structure and function. Binding of a ligand to an extracellular domain activates or inhibits this cytosolic enzyme activity. Duration of responses to stimulation of these receptors is on the order of minutes to hours. Metabolism, growth, and differentiation are important biologic functions controlled by these type of receptors. The most common enzymatic receptors: epidermal growth factors, platelet derived growth factor, atrial nucleotide peptide, insulin, and others, are those that have a tyrosine kinase activity as a part of their structure. Typically, upon binding of the ligand to receptor subunit, the receptor undergoes conformational changes, converting kinases from their inactive form to active form. The activated receptor autophosphorylates and then phosphorylate tyrosine residues on a specific protein. The addition of a phosphate group can substantially modify the three-dimensional structure of the target protein, thereby acting as a molecule switch. For example, when the peptide hormone insulin binds to two of its receptor subunits, their intrinsic tyrosine kinase activity causes the autophosphorylation of the receptor itself. In turn, the phosphorylated receptor phosphorylates target molecules, insulin receptor substrate peptides, that subsequently activate other important cellular signals such as inositosol triphosphate. Cascade of the activation results in the multiplication of the initial signals, much like that which occurs with the G protein coupled receptors. Now I move toward the fourth type of the receptor, that is intracellular receptor, which is considerably different from three receptors. In that receptor that is entirely intracellular, and therefore the ligand must diffuse into the cell to interact with the receptor. This places constraint on the physical and chemical properties of the ligand because it must have a sufficient lipid solubility to be able to move across the target cell membrane. Because these receptor ligands are lipid soluble, they are transported in the body attached to plasma proteins such as albumin. The primary target of these ligand receptor complexes are transcription factor. The activation or inactivation of these factors causes the transcription of DNA into RNA and translation of RNA into an array of protein. For example, steroid hormones exerts their action on the target cells via this receptor mechanism. Binding of the ligand with its receptor follows a general pattern in which the receptor becomes activated because of the dissociation from a variety of protein. The activated ligand receptor complex migrates or translocates to the nucleus where it binds to specific DNA sequence, resulting in the regulation of gene expression. The time course of the activation and response of these receptors is much longer than of the other mechanism described because gene expression and therefore protein synthesis is modified. Cellular responses are not observed until considerable time has elapsed, that is 30 minutes or more, and the duration of the response hour to days is much greater than that of other receptors families. Other targets of intracellular ligands are structural protein enzymes, RNA, and ribosomes. For example, tubulin is the target of the antineoplastic agents such as paclitaxel. The enzyme dihydrofolate reductase is the target of antimicrobials such as trimethoprim, and the 50 subunit of the bacterial ribosome is a target of the macrolide antibiotics such as erythromycin. Here I conclude my topic. For more information, like and subscribe my channel. Thank you.